certain amount for incoming students, a certain amount for people who are interviewing students, like PA, and then a certain amount that are like not part of the program at all, but PA.
Oh, okay.
At least that's what I understand how it works.
You know more than me. I haven't talked to Fisher this my first
I
year.
have talked to Fisher, yeah. And I'm looking forward to it.
She likes me for some reason. Don't know why.
Hey, don't put a gift horse in the mouth, I guess.
I'm oh.
Okay, are we missing something? Somebody? I think.
Missing a couple, but
Yeah, we're missing a couple. Okay. Guess we'll start nevertheless. Dr. Kai must have posted a message like this one over here. I don't know if y'all can see it. It has a file called the Laplacian eigenmap swissroll.m. So y'all can download that, and we'll be working on that on the first part. Should be on the Teams channel. And y'all can just open it up on MATLAB. It should look something like this. Okay. This should be on the lecture 12. Lecture 12, that should be the last message on there. Okay, so this contains a data set which is a nonlinear data set. So it's going, the decomposition of this data set doesn't really work really well. So we are going to try another approach. Like we have been working, you all have been working with PCAs, UMAP, and TSNEs. But for UMAP, there's a step that goes in before the UMAP, which generates a k-nearest neighbor graph. And here, with this example, we are going to run through how that k-nearest neighbor graph works and how we can use that decomposition to learn the underlying structure of the data. So if everybody has the data set, like the script up, we're just going to keep copy pasting stuff onto the terminal and seeing how it looks like. So first step will be to generate the data set. So just pick up this first whole part, copy it, paste it on the terminal. So what this does is that we are simulating a data set in this case, which looks like a Swiss roll. And if you want to visualize the data, just copy this part of the code and paste it again, and click enter. And that should generate a figure like this. So this is what our data looks like. If you go inside the data, you'll see that it has 3 coordinates, the x, y, and z coordinate, and we have about 2048 data points on there. And the data looks somewhat like this. Again, this was plotted using the normal scatter3 function. I don't know if I can make it any larger. But anyways, so this was plotted using the scatter3 function. If you want to change the color map, and if you don't like the rainbow colors, just set the C map. equal to, I don't know, Parula, and n is basically the number of data points you have. It was stored in that's how you started off here. This defined the number of data points you wanted to simulate. T was just a random number generator for each of these points. And this is how we set up as the T as pi variables, because we wanted to then put it into the sine and cos curves. So we turned our normal data points into radians. And if I just change the CMAP to Parula and then again run this, scatter, it's going to be a different color now. Okay, so we have just started off. So if you would just download this file over here, the Laplacian eigen Swiss roll. We'll wait a minute. That's fine. Just let me know when you have downloaded the file. Okay. Yeah. Okay. So now, as you can see, this data is not really a very linear or simple data set. So the issues we face with this kind of data when they are in like curls or circles or something like that is that we can't do a normal clustering-based approach to partition the data set. Like if you select in this case, if you select a cluster that wants to find the closest points from this point, a k-means clustering algorithm will catch, that's brush data. It's going to catch all the points like this. So it's actually going to catch a part of the inner curve as well, which is actually far away from the data we represent, that we want to put into our clusters. So we need to make some kind of transformations and only concentrate on the data points which are closest on this particular data structure. So that is why we are going to use the k-nearest neighbor approach in this case, because we don't want our k-means algorithm to shift from here to probably the center of the graph somewhere here. And that's going to make a mess. So you're not going to be classifying your data points correctly. So to do that, since we have 2,048 points and 3 variables, it's a very simple example. It's going to get way more complex if you work with actual single cell data. But this is a way to just make it simpler. So to run the k-nearest neighbor approach, we'll just be setting up these parameters, and the performance of your algorithm actually changes with these parameters. Okay, once everyone has set it up, you just have to copy paste this front portion of the code and send that into the terminal, and that should set up the data set. I'm just going to set my color map back to what it was, so it matches your data set. So this function is just the scatter3 function. I think this variable over here defines the size of the dots, and this is the color map. Okay. Are we set up? Are we set up to this point or no? Just asking. Like. Yeah. the files here in the Teams channel.
It's lecture 12.
Yeah, lecture 12. Yeah. Just download it and open it up in MATLAB.
Channel for lecture 12. It's in the hidden channels.
Yeah, I can come in. It's in hidden channels.
Yeah, I don't know why.
Why is it not hidden for me?
It's hidden for me for some reason.
Huh. Oh, I think I'm on last year's lecture. That's probably why. I'm on 2024, I'm not on 2025. Yeah, there you go. This is the hidden channels. Yeah, it's hidden.
So did you take the class last year, or
No.
you were TA last year also?
I wasn't a TA last year. I don't know. He just put me on the channel. I took the class 2 years back. Okay, yeah, so this is 2025. Yeah, this thing is hidden for some reason. Maybe he'll publish it today. I don't know if I can publish it. No, I can't publish it. Maybe I can. Anyways, so if you've downloaded that file, you should be able to load it up on MATLAB. Just, I think double click works, if I'm not wrong. Okay, so coming to the k-nearest neighbor approach. The k-nearest neighbor approach basically maps all the pairwise distances of all the points in our data sets, which are, so it's going to compute a 2048 by 2048 matrix. And by doing that, now we'll know how far away each point is from each other. So now we don't want to use all of that information, which we normally would have used for a k-means algorithm. We just want to keep the closest points, the top, say 30 or 60 or however much we want to set up, and just store those points. And this is what this part of the algorithm does. So if you copy paste this, in this case we are setting the parameters of how many closest neighbors we want to identify. In this case, I think the KNN is 62. If you want to go for a different number just to play around with it, you can. This is just a parameter, and it depends on your data set. So in this case, this is 62 because we are just capturing like 3% of the data, which 3% of the total data points, which is mentioned over here, the 0.03 star n. That's what it says. I'm gonna actually try to make this larger so that it gets captured on the screen better. Just a second. Oh, wait. I should probably do it. Yeah, so 0.03 into n just takes 3% of the links. So the next step is, once we have defined these parameters, we calculate the pairwise distances, and again, just copy paste this part of the code into here. And now what we have done here is that when we have the pdist function over here, which compares the pairwise distances, we sort the distances in an ascending order, and we keep only the k distances that we want to work with. So that gives us this NIDX matrix over here, which basically has the first row of it is just the points that we are working with, and all the subsequent rows are going to be its nearest neighbors. So the 10 is probably the closest neighbor of 1, 12 is the next closest neighbor of 1, and it moves on, and it moves so on. For each point, we have their closest neighbors stored like this, based on the pairwise distances. And this is the most important part, because in this case, we are reducing the whole dimensionality of the data, and like all the distances in the data set, but we are just concentrating on just the closest points in the network, and not in this k nearest neighbor network, which was from this data set. So in reality, what we are doing is that when we are selecting this point, we are just we are restricting our search to just this small, so this just the k nearest neighbors over here. So we are not letting our search algorithm go this far away. So that means we are going to just capture the local structures instead of the global structures. And this is very important when you're working with nonlinear data like this. So the next step is going to be to set up, again, this is going to set up the Laplacian matrix, and that's how we are going to represent this k nearest neighbor graph. So the Laplacian matrix is going to be a sparse matrix, so I don't know if you'll be able to see it. I'll show the exact structure of it. So again, copy paste the code into the terminal, press enter. It's that easy. Yeah, so the W matrix over here is a sparse matrix, but I can show the structure if you just save a new variable, maybe W1, no, full. Now we want to leave it in a sparse matrix format because it's way easier for the machine to handle, because it takes up a way lower memory space. But this is what the Laplacian matrix looks like. So what we have over here is that this is a normalized Laplacian matrix. So the diagonal elements of this matrix is going to be just ones. And each of these values where we have an interaction, like one has a 4 is the closest neighbor of 1, so we have that value stored over here. And this is going to be a symmetric matrix. So this normalized Laplacian matrix always has its diagonals as 1. Wherever it doesn't have any interactions, it has zeros. And wherever we have a closest nearest neighbor present, it has that distance information stored in there. Like how far is it? Like in this case, it's going to be 0.92 or something. Yeah. Okay, but yeah, so that is stored in the sparse Laplacian matrix. The next thing that we would want to do is just select the original normalized graph Laplacian. We are just storing it in a variable called D0. And we are ensuring that it's a symmetric matrix. That's just what this whole part of the code is doing. So let me just save D1 and we can look into what D0 is. Same way as you convert sparse matrices to dense matrices, let's just make it full. Sorry, it's DO if I'm not wrong. And this is what it looks like. Now the whole Laplacian matrix has been normalized and it has been made symmetric. It's just an operation so that we are reducing we're getting our values to a good level so that we can next proceed with a good range so that we can proceed with the eigenvalue decomposition of this. If we just did a straight up an eigenvalue decomposition of all the points over here, there's no point of doing an eigenvalue decomposition in this case because it only has 3 variables. We usually do an eigenvalue decomposition when we have way more features, but in this case we have way more data points and we have less features, but all the features that we have have a nonlinear relationship. So we have to go through a k-nearest neighbor approach in this example. So this part of the code just does the eigen decomposition. The LA parameter over here just says that we want the largest, we want the eigenvectors associated with the largest eigenvalues. So we capture the maximum amount of variance that we have in this k-nearest neighbor graph. Once we have that set up and the eigen decomposition goes through, just run this portion of the code as well. We can actually see what each of these eigenvalues consist of, the eigenvectors look like. Again, so this one is going to just select, this one is going to select just 2 random combinations of eigenvectors and it's going to plot it. So it's just a plotting function in loop. So yeah, so now what we have done is that we have changed our whole graph representation into a 2D space where we can actually look into their relationships. Earlier we were working with this. Now k-means approach is going to get confused on this graph, but now if you feed in this, the k-means method is going to be way more efficient because now the closest neighbors have been put together and now it's a more linear data set. So by doing this transformation, you're actually capturing the local interactions between the points and making your algorithm or classification task or anything that you want to do, like annotate cell types in your single cell RNA-seq data, annotate cell states. So this simplifies the whole process. And this is exactly what goes on in the back end of the UMAP algorithm. Computes it takes all the data points, computes the distances between them, selects only the k nearest neighbors, uses that k nearest neighbor, transforms it into a Laplacian matrix, uses that Laplacian matrix and decomposes it, and this is the decomposition. So now even if the interactions between your genes or features in your data set are nonlinear, the cells are still represented in a mostly linear manner where you can do your machine learning tasks way easily. So just the last portion of the code is just going to plot the original data and the nonlinear embedding together. Yeah, and should look something like this. So the k-means approach is going to be way more simpler in this case because now it knows, instead of having this Swiss roll kind of thing, we have somehow unrolled this whole Swiss roll using machine learning. Yeah.
That looks different.
It will be different.
Okay.
So the thing with that is that the eigenvalue decomposition again is a machine learning algorithm, so it tries to minimize to the lowest energy or the lowest solution. But the thing is that it all has a random seed. So if you set a seed, so I think you can set the seed as 0 and then you'll be able to replicate your results all the time. But if you don't set a seed, it's going to start from a random location. And in that case, it's going to give a slightly different solution, but it's going to be the same for any other algorithm at the end. Like if you use it for clustering or anything, it's going to still give you very similar results. It might be slightly different because it had a random start, but it's going to be the same. Okay, it's done. That was the last step. Yeah, so this is how you can work with the graph Laplacian matrices, and it's very handy to learn local structures when you have complex data sets like these. Okay, now I'll let Professor take over from here. Yeah. had the SciML workshop thing today?
Which
Damit?
was silent.
You were sent an email to us. Damit, they had a speaker coming with some vision stuff.
Oh, I was there. Yes.
Yeah.
Yeah. So
You mentioned.
yeah. So I will be there. Okay.
I haven't.
Yeah.
Okay. I have to go prepare for Dr. Sivari's class now.
Yeah. Thank you. Yeah, so later this week I will send out a survey to see, to ask you what the best time for you to do the presentation, because some of the students say, okay, I want to do on Tuesday, some say Thursday, so we have to vote and see what's the best time for the most of us for the last week. Then the finals, the exam, will be online and 30 questions. We still have to decide will be on Tuesday or Thursday. You want to have more time to read through slides, or you want to do this straight away. Okay, so I will send out the vote. So I think it's time to update this. If you know this orange button will help you if you have this shortcut here. So this is 4.5, the older version. So we need to update this. by checking the update. So after Shran showed you this Laplacian eigenmap with this example, we are going to download another package. So this package is developed for manifold learning and dimensionality collection of a series of methods. So what we have to do is just come to this page and then so this button is from this toolbox, so I just restart to update. So this is the latest version. We are not going to use this right now. Just close it. So we are going to download this toolbox. So Laplacian eigenmap is just one of them. So if you look at the number 10, this method, it's one of the methods which has been implemented in this toolbox. So there are a lot. So what we can do is download this, and then please click here to download. Okay. Now it's in my download folder. So I have to do right click, and then if you have this unzip program, we can do this extract here. So this is just step one. So you extract this GZ file into the tar, and then found this tar file, and then do this another round of extraction. So extract to this toolbox folder. You may have your own way to do the extraction, especially if you're in the Mac machine. But anyway, you can unzip this and find the folder. So that's the place where this toolbox is located. So now it's installed. We need to you need to copy the path. So we are going to add this path to the MATLAB, so MATLAB knows when we call this function, I need to search for this path to find this newly download function library. So I copy basically this folder name and come back to MATLAB. In here, I will do add path with a bracket, and I put my path name in there. If you don't do this, the function, the hundreds of functions under that folder will not be accessible. But now you should have this. What we can try is this toolbox provide a user interface. So this is called a DRGUI. I hope it can run. So DR stands for dimension reduction. So this toolbox is a dimension reduction toolbox. I hope this GUI can run smoothly in the new version. DR2. is DR2. I think we need to add a parse by I believe we need to add several subfolders under this root folder. For example, we need to add GUI. We need to add, so you can, when you tab to here, you can do the tab, and the next subfolder will be given here. So you can add this. Remember to finish the string by finish the bracket, the quarter. And then these two subfolders also been added into the parse. I think in this case, we should be able to run that correctly. Okay, let me check if you have this interface, and we can move forward. You can play with this interface yourself, of course, by loading stuff. Good. Yes. Looks better on your screen. Yeah, there. Good. Yeah, I think that's the GUI author provided. Yeah, you can load the example data and then play. It should have a couple of them. One more step. Yeah. Not so sure. Oh, you didn't finish the string. If you do this, finish the string. Yeah. Then drGUI. I think in the future, if you decide your data worth exploring using different dimensionality reduction technique, you can find a way to formulate your data and make them into a MATLAB readable. For example, you can put in the TXT file or this Excel spreadsheet, or you can find a way to make MIT. Anyway, so you should be able to load example data, for example. So we just try Swiss row again, and then you can load other your own data. But for example, we have this loaded scatter plot. Somehow not working, but that's the similar data set you have. Time series plot. Okay. And then estimate dimensionality. Basically, this will use those algorithms to find a number which is integer, should be as a hyperparameters to give to some of those dimensionality reduction technique. This is optional, so you don't really need. Then the computer mapping, we can select PCA first, for example. Then you have a 2D to map to 2D, and then compute it finished. Then you look at this is a PCA result. Then you can select Laplacian eigenmap you just had, and then there are other parameters. And this is the result for this technique. Isomap is done, then look at the scatter plot. Yeah, that's basically it. And if you want to know more, you can look at the source code of this and see how the individual function was called internally. So if you decide to do command line, you can do command line. Otherwise, this interface should be able to give you most of the results you need. There Tisney here, I don't know, it's its own implementation or using MATLAB based the built-in. Any questions for this part? It's okay. By the way, if I'm not wrong, this author of the toolbox is author of a Tisney method algorithm. Okay. Give it a try, Tisney method. Diffusion map. And you can also load the different example data. You load this, and then we will do the dimensionality reduction on this data. Okay, so on our agenda, next we are going to use a real example to show if you have a data set which you want to download from a geodatabase, because there are values to download this data to load into your own computer and to play with this data, although most of this data set has been published. Other people have already worked on those. But we want to repeat some of this analysis, or you can use your own method to find new insights, new results from them. So this data is from spinal cord injury mouse model. So we have data from a different time points with replicates. However, the software we developed for download data from this so-called sample level. So you mostly will need a accession number with a GSM. But if you click on one of the sample GSM access, there's no file under this folder. So we cannot do this sample level. What the author provided, they upload a combined sample file, which is given in this RDS file format. So what they did is they did the Surat in R package. They process all the samples together and give you upload this combined RDS file. So we checked those individual barcode file and the feature. They're not well prepared. If you open them, they are not a single file, and they put a lot of empty files in there. So they're not useful for our package. So what we have to do is to download this RDS file. Let's do this. It's a little bit bigger. It's a 1.8 gig. So we are going to download this, and then we are going to unzip it, because this is an RDS file with a GZ at the extension. So we need to do the unzip. Then I will show you, we should be able to read the information from this RDS file to MATLAB. And we are going to import the annotation, because they already done the annotation. So if you want to copy what they have already done, I will show you the way you can just put the information in, so we don't have to do our own cluster analysis. We don't have to do cell type annotation. We just copy everything out from this file. So let's download. It'll take 5 minutes or so. In the meantime, you can visit this paper published in Nature Communications. Okay, so this is the paper. That's the figure 1. So this is the one single-celled study. So basically, panel C is the main figure of single-cell data analysis. They have UMAP embedding, so we are going to import this. They have annotation of all the major cell types, so we will repeat this. We will get this into MATLAB. Okay. So this is an example of the special treatment. Remember, normally you can just give the individual level of the sample ID. Okay, GSM. Well, what happened? Yep. So I just restart my machine. Another thing you need to understand is to import this, which is R. This is the Surat package prepared R file, data file. We need to have the Surat package installed. So if you remember you have R installed, just try here. If you do library, that's in R, remember. We just check whether the Surat has been installed correctly. So if you do this, oh, no package, so we need to install it. So this is what I did. I did this, and then I just call Surrey library and make sure it's okay. So this is the preparation which is necessary in order to read that file in. Let me know if you are okay with this. You have this R installed. You have the library for the Surrey. So we are going to, from MATLAB, to call Surrey, use the Surrey to open that RDS file we download. Let Surrey to help us to read and give the information to MATLAB. So we have the construction of the UMAP other people published. Okay. Let me see, you have still installing. Yeah, good. You have that, good. You have that, yeah, good. So right, R, you have R? No, okay. No, that's all right. Okay, good. Okay. So from the downloading page, we should have that 1.8 gigabytes file download. So remember, you need to know where you download, where you saved this file. Normally, it should be in the download folder. Okay. We will do the right click to unzip it, so we can read that file. Yeah, I have some issues with the new version. Let's see what we can do. Do you have an issue with this running? I just cannot do it. For some reason, it's installed too. Yeah, for some reason, my killed my D. Yeah. don't know. Let me uninstall. Let me install this. So I don't know what's wrong with my installation, so I just revisit this page and install that toolbox again. Do you have the issue? Same thing? Well,
I have issue with RStudio.
RStudio.
When I try to install the zero, it doesn't get the session RStudio installed.
Oh, yeah, that's a normal maybe.
But
I,
it doesn't install it. Like it just shuts off and then.
oh. So let me I install this. And reinstall it. Install the previous and then try to reinstall it and see whether get back up. Yes, something wrong with the package.
That wasn't working in this? Like after you were trying to load another like file in, or
Which one?
like what wasn't working?
The previous version is working, but now if you click this, it doesn't work.
Yeah, I just usually just call it to do.
Yeah. Yeah, I need to find a way to fix this. Yeah, so it's a problem with this button here, but if you do clean, well, this button makes things worse. I don't know what's wrong. Yeah, it looks like the problem was from this button here. But if you run after fresh installation, this should be still. Mm-hmm. You have the same problem. So what I did is just I installed that orange button, then use the command line. But still, yeah. So you still have this orange button. So let's do this. Just I install this, and then from here you just tap. Yeah, I don't know why that button caused a problem. Yeah, yeah, I'm sure. But for today, we just uninstall that orange button, and then yeah, can be not valid. Yeah, it's interesting. Yeah, interesting. Do you need to log in to get this? I don't think. So you don't have.
I just uninstalled.
I installed the whole thing. Yeah. So nothing here right now.
No, there shouldn't be anything noise in this directory.
Mm-hmm. Okay. Then. Temporarily unavailable.
Let me try restarting MATLAB, see if that helps.
Yeah, I never seen this error. Yeah, so what I found the problem was from the shortcut, but this should work right now. Let's try. So we prepare like this to load, then we take a break, okay? So let's try, first go to your download folder. We need to find this file. We need to unzip it, it's the big one. So you can do extract here, okay? Okay, you should have this, and if you search by the size, so I just file size, it's huge, so you should have this, okay, in your folder. So that's the target we are going to read, and I hope we can import, then find RDS file, and then to search for in the downloads, find this RDS, and give to so we need to have this R installed at the back end. So now it's time to take a break, and I will help you if you still have the problem. Okay. Yeah, so I identified the problem was the link with this orange shortcut. I will fix that later. But if you run the interface by itself and then load that RDS file, you should be able to get this. I don't know who get this already. You get, okay, good. That's awesome. It's not easy. You can see through the communication between two languages and the interface, there are a lot of details in there. So now the only problem is not all the information has been read back. We don't have the annotation. We don't have the cluster ID. Yeah. So it's good. So I talk about this. You don't have that installed. That's good. So you should be able to run if you have. Do you have the SG? Mm-hmm. Yeah. So if you can. If you run. If you have this installed somewhere. Mm-hmm. Mm-hmm. Yeah, you can use it. Mm-hmm. Yeah, so you can use this. It's similar. Okay, do you have this? So we are moving towards the next step. So this is if you check the gene, it should be okay. So the gene name should be imported. If you check this, select name of genes, it should be here. By the way, this is a very, very large data set. But if you see, I want to label this by batch ID, there's no batch ID. This is one. If you see, I want to label it by cell type, it's not there because the information in the R has not been correctly read into here. But if you check your workspace, there's a table called metadata. So this is read from the R file. So we just need to find which column is the cell type. Because every lab prepared their R package results called differently. So this lab decided to call this cell type annotation as a course clusters. Then you have all the cell type name. So there's no way for program to know which column cell type information should be imported. There's no way you can guess. And then we need another information is maybe it's original identity. So this tells you, or maybe it's a condition. So we need a condition as a batch ID, and then need this column as a cell type. That's the information we need. So what you can do is now to save this into the workspace as SCE. So we need temporarily save the information in the workspace here. Then from here, we are going to do manual annotation import. So this will be C underscore cell type. So we will assign this metadata variable which called and make this into a string. So this operation, it's a long command, but that's this operation will extract that column of string that gave to cell type. Yeah.
I don't seem to have the metadata table in my workspace.
Yeah, so because you use older version, this crash version will give you this. Yeah, but I will give you the non-crashed version and with this. Okay. Yeah. So if you don't have a metadata because you're using the previous version of MATLAB toolbox, it's okay. So next update, working version, you should have that. So this will help you to import the cell type. Now we need the batch ID. So batch ID. So this will we will need string metadata table dot then conditions. We know it's a condition. So now we have the essential information we want to, the clusters which annotated by the authors, and the cell type, and also batch ID. They know which cell is come from which sample. Now we need to load back the SEG. Okay. You can also do from here. So you can see, okay, I can read, import data. The current one I will remove by replace with load, import this from workspace. So this should be found this and read back. Now this has been updated. Now you should be able to look at the cell type, and this is from the original paper annotation, basically, because you copy that information in there. Okay. Mm-hmm. You miss a step. Do you have? You don't. You have. Do you have the? Yeah. So you have this prepared.
I don't think this one prepared though.
I think it's okay. Yeah, you're prepared. SE. Yeah, it should work. No, I think you haven't. Yeah, so you go back to the interface. Yeah, and then you do export. Let's save this. Yeah, save this. Okay. Get rid of one. Yeah, just override. So yes, then do that two step again. Mm-hmm. Then, yeah, then you load it back. This should work. Then the yeah, so you should have everything the same as the paper. And you can save this on your hard drive. So next time you just load it back. Yeah. So you have some problem? Okay. So let's see. Do you have SE? Okay, it's here.
I thought I have some.
Mm-hmm. It's a yes. Mm-hmm. And then from workspace, yeah, load back. Say okay. Now you can cell type. No, yeah, next one. Yeah. Mm-hmm. Cell type. Yeah. Yeah. So you can. So basically, we get this information back from the paper, and then you can do whatever you want here. Yeah, I know. Yeah, but yeah, it's okay. You have a video recording for the class. Yeah, you didn't save this. Do you have the interface? Yeah. You do export. Yeah. And then okay. Get rid of this. Just do SCE. No, check. Put a name SCE. Yeah. Mm-hmm. Just override it. Mm-hmm. Yeah, then you come back to here to do that 2 lines. You don't have to copy, you just use up down. Yeah. Do you have metadata? You may not have this. Okay, you already. didn't use the right name. Yes. Yeah. Okay, so this is this part, and we are done. So you know how to deal with this situation. In geodatabase, you should find quite a lot of those people upload RDS files. So this is the way you can get the data in. So let's check the batch ID. So we have yeah, so they are very, very well mixed. What we can do is to batch ID, then see how many different samples. They have samples from uninjured, then 1 day, 1 week, 3 weeks, 6 weeks. So those are the samples. Looks like their samples, everything just overlapped onto each other. I believe they used some of those alignment or harmony stuff. Yeah. But that's the data they provided. So remember to save this, so next time you don't have to do that again. So but now you have all the data. Okay, so we can move to next. We should be easier for you to load example. Load example data. It is smaller to play with. Okay, so this small example data, what I want you to do is to export this variable SCE and also gene name and X the matrix. Okay, if you click this X, you should see this is a highly sparse matrix. If you look at the genes, the first gene is this, the second gene is this. What we can try first is to do a scatter plot, for example, between these two genes, number 4 and 5. So that means I'm going to extract row number 4 and the row number 5 and to do a scatter plot. So my row number 4 is X4 is from big X, you extract number 4, the row, and your X5. Right, so 2 genes. The first gene is ATP6, V1H, the second 4, 5. So now I want to know whether these 2 genes are co-expressed. So what we can do is do a figure and a scatter plot. X4 versus X5. If they're positively correlated, then the cell with a high expression of X4 should have a high expression of X5. But what you can get is something like this. It's very useless because the sparse the matrix and all the values. Integers, there are maybe a couple of cells with a 3-3 expression, several cells with 3-4, so give you a point, but most of others maybe 0-0, okay? So you get the idea. So if you look at this x4, you see the expression x5 looks like this. So that's why you get a scatter plot which almost tell you nothing about the correlation between these two genes. So that's where the imputation may comes in to help you. Now we are going to use the method we call the magic, okay? It's a manifold alignment, manifold learning algorithm. So we can do run dot. This is a MATLAB based ML instead of MATLAB magic. So if you give magic the X and ask it to give me a imputated version, impu, remember to put a semicolon at the end. So the output of this imputation method will give you another matrix which have a same dimension with the original X, but X is sparse the matrix, but the imputated version will be not. So this is the same imputated version of X, okay? Now if you get, remember this is a command line, so if you still need this, just copy that. You can name this as a X2, you don't have to be X impu. As long as you know which one is original, which one is after imputation. So with this imputation, let's try to get a row 4 from imputated version. And row 5, instead of original, we do this imputated version. Then from the scatter plot, now you see this relationship between two genes. Looks like these two genes are somehow positively correlated. So if you have a cell have high expression of first gene, you should have a relatively high expression in the second gene. So this helps you using manifold learning approach to learn cells, genes from each other. Remember this data contains a lot of different cell types. So this region, so if you look at our cell type, we have different cells. So maybe this group of cells are from one cell type, those from another cell type. It's possible. So yeah, probably we should try, see whether the cell type play a role in here, right? So I can give the size and then SCE cell type. Scatter. So I'm just trying to see whether me see how to use this function. So I want to use this scatter plot to plot different colors. So you can see the color indicated the cell type. So I will see how I can do it. So we need scatter plot, the size and the color. Okay. Size is 5. The color. Yeah, it looks like they're indeed the different colors and indicate a different cell type. So this relationship between these two genes depends on which cell type they come from, those cells. Okay. So let me make it bigger. So this number is the size of the marker, and we convert this cell type into a categorical variable. So the program knows, okay, I can use that name as a different class of the points. Okay, so this is what I got from. Just in case you need this again, so this gene number 4 and 5 are just two random genes I randomly selected. But if you have some target, so you really want to know the relationship between these two genes, this really help you, and I found it informative, and also the cell type information will be color coded in this way to help you. Do you have that? Not yet.
This didn't run properly for me. It took a while.
I think you are done. Yeah, this very quick is already done.
But this is how it came out.
Oh, the error message.
I think I didn't I probably missed a step where I didn't.
Oh, yes, maybe in your case, let's do. Okay, in your case, if you have the errors, do a function called for, and this is x for x, because your x may be a sparse data set, may have some impact on your imputation. Do you have that? You don't have enough memory to. Oh. Which x you are using? X. Why you have so many? Yeah, your data set is too big. I use small example data, so.
Okay.
Yeah, so I think you are using the one you just downloaded from the injured. Yeah, that's a big data.
Yeah. Should I
You need a better computer to handle that imputation. Yeah. So the one I'm showing you on the screen is a small example.
Could you show me how to do that real quick?
Okay. So for that, I just load the SCE. Where is SCE? So you just load example data that will be over. So this is simple.
Oh, okay.
Yeah, then you just write this to overwrite the.
Oh, I thought you
Then-
meant taking out just a couple of rows, or like just taking out a batch.
No, I just change everything. I know it's too big for
Yeah.
for average computer. Yeah. Oh, you cannot see the end. See, you just the rule is you just tab. Okay, this should work. So your SCE is different from the, so make sure you have the right SCE. Your SCE probably is original.
Yeah, because.
Yeah.
Okay.
Yeah. Yeah, I use the old one. Yes.
I also have
Yeah,
to
you
set it.
just reset. I see. That's fine. Yeah. Okay. So that's the imputation you can use. Yeah. Scatter plot. I don't know. I don't know. Yeah, that's, I don't know what's happened. So, it looks all right to me. I don't know. So another place I think you should use imputation is when you do a violin plot. So violin plot assume your data is continuous variable, and so you have average, you have outliers, you have just like a box plot, you have 75 percentile, 25 percentile. If your data is only 0, 1s, 3, just integers, we will not give you very informative distribution information. So that's why, let me show you the examples. So now, if you want to know one of the genes expression across all the cell types, you can see I want to do this violin plot. So the violin plot is here. Okay, this button here. And then you want to select cell type and then include them all. And then you want to show the gene expression, then select some genes. You can just paste random genes. Now, the dialog, I don't know if you can follow, but maybe I'll show you later. Now, if you don't do imputation, you just say no, then you violin plot, because the zeros, the violin plot, most of them will be compressed down to 0. You may have some high-level expression from several cells, then give you the expression of the outliers. So this is the typical violin plot you will have. I don't have a zoom in, but you will get something like this. Okay. However, I encourage you to do the imputation when you want to do. So let's do that again. So click this button and select cell types. I get rid of some of these long names. Okay, I only select those ones. And from here, I select gene expression for select genes. And then from the paste, there's a random genes. There's too many, I just select few of them. And then in this step, I will do a imputation with magic. You can plot them in separate, but we can try again with the same figure, put everything together. Oh, something wrong with this. Yeah, do that again. Cell type done. expression, paste some genes, do imputation. Plot on the same figure. So this imputated version really provide the meaningful mean and distribution of the expression. So you can see the shape of violin, okay? So there some kind of bimodal distribution, some outliers. Okay, yeah.
I keep figures that are similar to the one you first just made.
Mm-hmm.
Am I just not manipulating this properly?
Yeah, you just click this to zoom in. No, maximize this table. Yeah.
Oh, okay.
Yeah. That's beautiful. Yeah, so you just click this to maximize. Yeah, there you have this. Mm-hmm. Okay. Yeah, so I encourage you to use this for most of the case. This should give you, if this gene really express high in this cell type, you should have a clear separation. If you don't do that, everything will be in around 0. You don't see that. So this is just for visualization purpose, and make sure the consistent with your biology stories. Yeah. So we still have 10 minutes. I will show you the tensor. So if you do okay. So you know you can easily to create 2D matrix, okay, if you do the random and the 3x4. So that will give you a matrix, okay. You know you can do SVD with this, SV. This is just very normal operation. You can also create a multi-dimensional matrix. So this matrix is 2D. Then, but if you see, okay, I want to have 2 layers of this. So basically, you have a first matrix is 3x4, and behind this you have another 3x4. So this will have 24 numbers, random numbers in this qubit. Not a qubit, it's a block. You can get the first x So this is the first layer, okay? Then behind this, you have another layer, depends on how you see it. So this, so X is not a matrix anymore, it's a tensor. You can do even higher dimension if you want to do another layer, so 3. So the fourth dimension, you can put even 5, another. So this function, for example, this random number generator, allow you to generate the structure of data with a high dimensional array or tensor structure. Now the problem is a lot of other functions. If you have a x is 3 by 4 by 2, and now the SVD will not work because SVD is a function which is not designed for deal with tensor structure, and it doesn't know how to handle this 3D stuff. So that's why we rely on third-party tensor-based toolbox. So this is a place where we can download a toolbox. We can quickly do it. Current version, so you know how to do it. And download the source code, and in the folder of downloads, right-click and let to extract here, and this is the folder. Found the folder. So you know where it is, then you need to copy this parse and add a folder, add a parse. Now I just installed Tensor Toolbox. Now we are ready to use the function in this toolbox to deal with a data like X, which is a higher dimension, higher than the matrix dimension. So the function we can use is called to do a tensor. Okay, so we make this let's see, X. I don't know how, but it's 3. Yeah, some issues with this. Only normal. Yeah, I don't know where to get this Z. Let's do this Z random, make a bigger one. Let's see, prepare a tensor Z. This Z have a 25 by 25 by 9 random number. Then we make a tensor Z. Here I probably have a typo. I don't know. I can try, see whether this works. If not working, I should put TZ in there. Maybe. Let's try Z directly. Yeah. So let's do that again. Let's do that again. So I had a typo in there, so that's why. So TZ, so we make this MATLAB tensor into a tensor which can be used. And then to do this CP decomposition, we put a TZ here. This should work instead of Z. So this start to decompose, okay? So let me make change on this page. Instead of Z, we need a TZ here. And then after decomposition, we can get back the so this is finish the decomposition into this. Okay, 3 major components. Then we can put them back. So this will use these components, reconstruct the tensor, and then get back Z. So remember we had a Z 25 by 9, and then here reconstructed. Also share the same structure, same dimension. But this is reconstructed after decompose and pick top 3 and put back. So this Z data is the denoised version of original Z. So this is the whole process. You decompose the tensor into components, then merge it back. The same idea as VIDI low rank approximation. So this process is useful to get rid of noise of small variation in the original Z. If Z have a major structure in some dimension, this process will learn and give you the better or clean version of that. Okay, I think that's all for this week. Any quick questions? Okay, yeah.
Not related to this, but for the homework, I was trying to upload some GSM files into the Singapore GA tool. I was getting an error about the index being larger than 0.
Okay, so send me the procedure repeats, and I can repeat this error and fix that. Sure, yeah. Okay, see you next Tuesday.